Good day, everyone. On behalf of BioIT World's Global Web Symposium Series and our sponsor, Agena Bioscience, I'd like to welcome you to a pharmacogenetic PGX Blindspot, the prevalence and impact of CYP2D6 hybrid alleles. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I'd like to introduce our presenters for today. First, First is Dr. Houda Hachad, PharmD, Chief Scientific Officer, Translational Software. And our second presenter is Dr. Daryl Irwin, PhD, Senior Director, Scientific Affairs, Agena Bioscience. Welcome, Dr. Houda. The presenter ball is yours.
Hi, everybody. Thank you for having me today. Thank you to Daryl for inviting me and the Agena team to providing this talk regarding CYP2D6 hybrids and the complexity around their detection. So during this talk, I'm going to briefly do a small introduction of our company, Translational Software. And then I will talk about the tools that we have for pharmacogenomic implementation. And I will choose CYP2D6 and the testing for CYP2D6 genetic variations as an example of a complex pharmacogene, but yet very interesting and relevant for clinical use. I will briefly talk about the solutions that exist for CYP2D6 testing, highlighting their strength as well as their pitfalls. And finally, I have prepared a couple of case studies that can show you how sometimes if you don't use the proper technology, you can actually miscall or the right diplotypes and therefore assign a wrong phenotype and a recommendation. So before we start on CYP2D6 per se, I want to talk to you about translational software. So we are a technology company located in the Seattle area, and we have a platform that has different technologies that allow us to work with different parties that are interested in implementing pharmacogenetics, namely diagnostic laboratories, but also clinical organizations or other groups such as pharmacy benefit managers or employers. And what we do is we have a set of tools that allow them to get quickly to the market and also to scale their business properly. We have tools around interpretation, but also tools that can allow us to do population data analysis and identifying cohorts that are likely to benefit from pharmacogenetic testing. We have been in business since 2013, and we have learned a lot in implementing properly pharmacogenomic programs. We have been very successful, and we have covered testing and result interpretation for millions of patients around the world. We operate mainly within the United States, but we also have customers in Europe, in Africa, in Australia. Our results have been reviewed by over 32,000 physicians, and our technologies are integrated with a number of existing software systems that are embedded in existing workflows or other software systems that are used in hospitals. We have a number of criteria that allowed us to start with pharmacogenetics. And the first one is that it harbors excellent evidence. As you know, the FDA and actually pharmaceutical companies have been sharing information about genetic variability for many years. And this information is communicated in the FDA prescribing labels that every drug has. And this data has been available to doctors for many years, but they were not able to act upon it because of the limited testing. When testing became widely available, this presented the opportunity to now using this information more broadly. The other criteria for pharmacogenetics is, as we know, many people are medicated, and actually a number and a large proportion of the population, especially in the United States, takes more than 3 medications. So this gives us the opportunity with one test to inform multiple therapeutic strategies and help optimize this treatment for these patients. We are lucky within the pharmacogenomic world that we have a community that have coalesced around a consortium here in the United States called the CPIC Consortium, but also other groups outside of the United States. And these experts are looking at the evidence and producing evidence-based guidelines to allow practitioners to access and to act upon this information very easily. Finally, the pharmacogenetic data is data that can be used perpetually and across a lifetime for a patient. So one test conducted early can allow us to reuse this information multiple times depending on the needs as they change and as this patient develops diseases or comorbidities. One of the characteristics of pharmacogenetics as a discipline is that it requires heavy understanding of not only the genetic aspects of these genes that are tested, but also the clinical pharmacology profile of the medications. And it also requires technology and the ability to weave this genomic information with the pharmacology data points in order to produce this actionable report for that physician. And the relationship between a result from a test and a recommendation is not straightforward, where you have one genetic trait that will give you a certain consequence. Often, for some complex situations, you need genetic results for multiple genes to be able to inform the guidance for medications. And for the same gene, if you are interrogating for many variants, you need to look at the collection of the variants that are harbored by an individual in order to predict a phenotype. And a phenotype cannot be translated for a class of medication. The relationship is between a phenotype and a unique medication. So it requires a level of scrutiny that is very delicate, and it allows us also to keep up with the literature and see how evidence changes. So at Translational, we do multiple things with our technologies. We allow a lot of entities to actually optimize their panels and the tests and the variants that are selected for interrogation. This is a very important step because it allows us to ensure that the proper variant in a given population, especially here in the United States, which is a multi-ethnic population, all of the variants that are relevant are included. It allows us also to discover the technology that this entity has chosen to use for testing, and also allow us to identify the limitation in that technology. What are the genetic signatures that can be revealed and those that cannot be revealed by the technology selected. Once this panel is optimized, we work closely with our laboratories and getting the PGX results from any instruments in the native file format that is produced by that instrumentation. We then proceed to standardize these results into the same representation, which will allow us to then produce this genetic type from this collection of variants we have observed within a patient file. The genetic typing requires complex operations such as haplotyping, because for some pharmacogenes, and we're going to talk about it for CYP2D6, the interrogation of multiple variants, whether sequence variant or structural variants, will allow us to infer the likely haplotypes for that individual. And finally, once we have that genetic type that can be expressed in terms of sets of haplotypes, which we call diplotypes, we can then overlay a predicted or a likely phenotype, which then will allow us to retrieve the recommendation from these guidelines and map them to that particular pharmacogenomic signature for the patient. And finally, We have a technology that allow us to do custom reporting and custom distribution through our API of those pharmacogenomic signatures in order for us to embed them into existing EMR systems or records for the patient. The tools that allow us to implement successfully pharmacogenomics are the availability of multiple platforms to choose from. They're not all equal. Some of them are more powerful than others in detecting a large set of alleles and complex structures such as these hybrids. We have the advantage of having small groups that are very focused on keeping up with the evidence and the emerging data around pharmacogenomics, namely PharmJKB, which is the largest knowledge base that collects the annotations and annotates every association. But we also have, and we are participant of PharmVar, which is the largest catalog of pharmacogenomic alleles, especially for the cytochrome P450 enzymes. We also have a number of reference materials that facilitate the validation of the tests for the laboratories, and these can be accessible very easily. Finally, as I previously commented, we have these strong consortia that are providing us with recommendations alongside with the FDA prescribing labels. And we also have a focus expert working groups that sometimes look at difficult topics or complexities such as genotype to phenotype relationships and issue recommendations for the community as a whole in order to standardize testing. So for CYP2D6 per se, there are a number of focus groups that have been operating within the community that looked at this particular enzyme because it is one of the major pharmacogene and the most studied pharmacogene. It has been studied since the 70s. And this particular enzyme gene encodes an enzyme that belongs to the drug metabolizing enzyme family. These enzymes are very important in the clearance and the elimination of many medications. And by metabolizing these drugs, the levels of the drugs as well as their metabolite can vary, and therefore it presents an opportunity for us to test for variability through genetic testing. The enzyme itself is localized mainly in the liver, where it plays its main role as a drug metabolizing enzyme and governing clearance. But it can also be found in the intestine and the brain, although we don't really understand clearly its effect in these organs. The enzyme matures very early after birth, and after a couple of weeks, babies or newborns have levels that are comparable to adult levels. And therefore, it presents an opportunity for us to allow testing and genetic testing earlier in life, and we can keep that data and store it and reuse it over the lifetime of an individual. Finally, the enzyme, although it's a drug metabolizing enzyme, it is also known for metabolizing other compounds such as endogenous substrates, but also toxins that can be in the environment, such as pollutant. So it has a main role of detoxifying enzyme along its role as a drug metabolizing enzyme. And finally, the enzyme itself is involved in the metabolism of over 25% of commonly used medication for a number of diseases. CYP2D6 has by large the largest number of guidelines associated with it. This slide has a list of the CPIC guidelines, mainly for tamoxifen, which is an anti-cancer medication, but also two guidelines that are key for behavioral health medications and widely used, namely tricyclic antidepressant as well as SSRIs. These guidelines have been issued since 2013, but and they are currently updated with a cadence over every 2 years as new data emerges. The slide has examples of medications that have guidelines with them. As you can see, a number of psychotropic agents are listed. These can be used for depression, but they can also be used for anxiety or other psychiatric disorders such as schizophrenia or OCD. In the medications also that are heavily used for pain, such as opioids, are substrates for CYP2D6, and their efficacy as well as their safety profile can be heavily affected by genetic variability of CYP2D6. In cardiology, we have a number of antiarrhythmic medications that are metabolized by CYP2D6, and testing can inform us regarding their safety. In neurology, we have medications that are not widely prescribed, but they are very interested. And for some of these medication, testing is actually required prior to prescribing the medication. So when CYP2D6 enzyme activity was first discovered in the 70s, genotyping, of course, it was not the genomic era yet. And the CYP2D6 activity was actually revealed using phenotyping, which involves the administration of a single dose of a medication, which is a cough medication called dextromethorphan, very old medication. We used to give it actually to individuals, and then by measuring the molar concentration of this dextromethorphan and its metabolite that's formed by CYP2D6, we would be able to classify individuals into those that have a normal activity versus those that have a reduced activity or a high activity. So we were able to build these histograms based on these drug to metabolite ratio, and we were able to differentiate between these two different phenotype groups that we called ultra rapid, normal, or actually extensive, as well as intermediate and poor metabolizers. And this is an example of histograms and how the activity fits nicely within this log distribution. In the 80s, when the enzyme and the gene was cloned, and we discovered that there are a number of genetic variants that are very common in population, we were able to correlate genetic variation with the observed phenotype. And we were able to establish with this activity score continuum a genotype to phenotype relationship and correlations. And by having a genotype for an individual, we were able to predict the likelihood that the patient would have a certain phenotype. We later developed a method of activity scoring by looking at the alleles that are revealed by the test and classifying individuals within this continuum between above 2.5 based on the score to 0, and assigning this genotype predicted phenotypes between the ultra rapid, to the extreme at the other side of poor metabolizers. And poor metabolizers are in the groups that are usually where your cutoff for that ratio is 0.3, and these individuals are predicted to have almost no CYP2D6 activity. At the other extreme, we have individuals that harbor very high capacity to metabolize and have a higher enzyme activity, and these are these extreme of this ultra rapid metabolizers. And in between, we have these two groups, the normal metabolizers that we used to call the extensive, as well as the intermediate, with actually a large overlap within that ratio, which makes it difficult in some circumstances to differentiate the actual consequence on a medication. So although we see these phenotypes as grouped into these four major groups, as you can see, it's a continuum of activity, and you can have overlaps between individuals that have actually the same ratio, but can be either a normal or an ultra rapid metabolizer. So this adds a level of complexity when we do a task to actually predict the likelihood of a phenotype. There's four main categories of phenotype that we can identify when we measure them either with dextromethorphan or when we do genetic testing. Their frequency is very variable across the ethnicities. And as you can see, some ethnicities harbor at a high frequency the ultra-rapid metabolizer traits, such as Oceanian, Near and Middle Eastern individuals. But the ultra-rapid metabolizer is not a common trait in populations such as Asians, for example. In Asians, the trait that is common is the actual poor metabolizer, which you can see here with this high percentage up to 6%. So it is very important to take that in account when you are testing in a multi-ethnic population. The CYP2D6 gene itself is a small gene, and it sits in a loci with two other pseudogenes, the CYP2D8 and the CYP2D7. And these are very homologous to the CYP2D6 gene, which requires careful attention to not do this co-amplification when PCR is done. So this requires optimization of these probes when you are conducting testing. The CYP2D7, that is the neighboring gene next to CYP2D6, has a T insertion, which makes it completely defective and is not expressed. Actually, the protein is not expressed in humans. And there are regions, despite the homology, where there are regions that can be actually used for specific detection of CYP2D6, where there are differences. One of them is this long spacer after the CYP2D7, but also the exon 9 regions are quite different between the 2D6, 2D7, which allows us to detect 2D6 more easily. In terms of the different variations that we know of for CYP2D6, there are two major groups. There are sequence variations, which are mainly SNPs, so single nucleotide polymorphisms, and also small deletions and insertions. And these, depending on the majority, are not complex for detection. The difficulty starts with when we want to detect structural variations of the gene. The CYP2D6 is known for harboring a number of complex structures, such as copy number losses, where your entire gene is completely deleted. But we can also have a copy gains, and we have multiple duplications of the gene, multiple units of the gene, as well as duplications or multiplications. And finally, because of the neighboring gene, we have complex rearrangement structures with CYP2D7 that can give us these hybrid structures. And the hybrids can be of two types. They can have the upstream region of a CYP2D6 with a downstream region of a 2D7, or the opposite. The upstream region formed by a 2D7 sequence, as well as a downstream region with CYP2D6. And these structures can exist as single units or single haplotypes, but they can also exist as tandems of these different, along with other alleles. So this makes this detection very complex. And all combinations of all of these structures can exist in patients. The largest catalog of these alleles and hybrids is, of course, PharmVar. Most of you are familiar with this website. There used to be the CYP allele nomenclature page, which transitioned to PharmVar 2 years ago. PharmVar is the largest catalog that allows us to discover the different alleles that have been described, as well as the variants that are used for their definition, and also the level of evidence that we have for those particular sequences. This is the tool that will allow us to keep track of new alleles and also allow us to discover the limitation of the existing tests that we might have. The PharmVar has a committee that I participate with for CYP2D6. We review these submissions of existing or novel alleles every month, and we vote on whether these are accepted to be cataloged within PharmVar, inviting implementers and assay developers to improving their testing as these alleles are discovered. The prevalence of these alleles, although is not available on PharmVar, can be available on PharmGKB or within the CPIC website. And as you can see, their prevalence is again different depending on the type of alleles, whether we're looking at the frequency of deletions or duplication. As mentioned previously, Asians are known to harbor by far the largest percentage of alleles with reduced function. If you compare them to Africans, where in African we have by a high percentage of the population that harbors actually duplications of alleles that can give that ultra-rapid metabolizers. And of course, in the United States, given the high frequency of these diverse ethnicities, the testing needs to be optimized to cover all of these alleles as well as the deletions and the duplications that can be identified. So I'm going to spend a little bit of time talking about different structures and the complexity associated with their identification. And the first structure is these duplications or multiplications. So these describe situations where you have either two, and other term duplication, or more than two gene units that are inherited on the same chromosome, and these are usually identical. So in this graph, we have on the first cartoon, two gene units that are inherited, and therefore this individual will have two copies of a normal function allele, which is a SAR2, and that can result into an ultra-rapid metabolizer trait, for example. An individual can inherit alleles that have no function, that are defective, such as the STAR4. And this is represented here in this second cartoon. But we have to remember that sometimes we have more than just two copies that are inherited. Individuals with up to 14 copies have been identified so far. They're very rare, but it can be identified. So a proper characterization of the existence of duplication is important, but also allowing us to know the copy count of how many copies we have is also relevant. On PharmVar, you have a comprehensive description of the number of haplotypes or alleles that have been described so far to harboring these duplications or multiplication events. And as you can see, normal function alleles such as the SAR1, SAR2, SAR35 can harbor this structures, but also decreased function alleles such as the SAR17, the SAR10, SAR29, or 9 that are known to be duplicated. The STAR41 is the only allele that is known to be duplicated but also multiplicated by having 3 copies. And finally, no functional alleles such as the 4 or the 3 are also can be duplicated. So, and because of the functional consequence that are variable among these alleles, the presence of these extra copies can translate into an alteration of the enzyme activity or not. This is a summary of examples of alleles with this activity scoring method that allow us to look at the characteristic of the allele and assign its function. And you can see that, for example, the no function alleles are not only the single units, the single entities of a diplotype with single units such as the STAR3 or 4, but they can also have multiplications of these alleles. So this can get very complicated when you are testing for these alleles and you are doing structural variations into properly characterizing the copy count, but also the type of alleles you are testing. The complex structures that we want to also cover today, in addition to duplications, are these hybrids between the CYP2D6 gene and the neighboring CYP2D7 pseudogene. In this cartoon, I'm looking at the STAR36, and the STAR36 is relevant because from a sequence variation point of view, it shares with the STAR10 the two amino acid changes that characterize the star 10, which is the 100C2T and the 4180. So two amino acid changes that if you detect them, you will call this particular allele a star 10. But if you look carefully at this particular hybrid, especially in the exon 9 region, downstream in exon 9, we discovered that there is a region that actually is no longer a CYP2D6. It's characterized here in red. It's actually a 2D7 sequence. And because of the presence of this conversion, this renders the 36 completely defective, and it's a no function allele. So unless a test can detect this exon 9 conversion, we will default to a star 10, and that will produce a miscall. The star 36 is complex because it can be found as a single gene unit by itself, but also as a tandem configuration. A tandem is basically two gene units that are not identical that are inherited on the same chromosome. And you can see it is on the 3610, but you can also have duplication of this hybrid, or you can have a duplication of the 10 along with 36. So multiple different complex configuration that can only be revealed if your test is allowing you to interrogate multiple regions and confirm the presence or absence of these hybrids. These hybrids, the frequency of these hybrids is well known in Asians, and as you can see, the 36 type of hybrids, whether in a tandem or as a single unit, are identified in Asian populations. And in some of them, such as the tandem for the 3610, is actually more frequent than the 36 as a single unit because it can be found up to 25% in Japanese, Chinese in Hong Kong, as well as Koreans. So it is very important when testing in these populations to ensure and properly call for that particular haplotype. The star 36 by itself as a single gene unit is also found in Asians, although at a lower frequency compared to the tandem by itself. The same hybrids, the 36 hybrid as well as its tandem with the 10, is also has been recently discovered to be also prevalent in African Americans. And this is based on two studies that looked actually at the presence of these entities in the United States in patients who were receiving pharmacogenetic tests here in the United States. And up to 1% of African Americans can actually have this 36 no function allele, and then its proper characterization becomes relevant this time not only in Asians but also into this particular subgroup. Another allele that also harbors the same signature, which is this exon 9 conversion to CYP2D7, is the star 4N. The star 4 is a subtype. The star 4N is a suballele of star 4. It has the splicing defect that characterizes it as a star 4, so it's a no function allele. But in addition to that, it has this exon 9 conversion here in exon 9, and that can interfere with testing. So although detecting for it is not going to fine-tune the functional effect, but it can actually interfere with the proper interpretation of the results. A recent survey looked at this particular 4N and discovered that actually it exists in tandem with the star 4 into up to 1.5% of these samples that have the star 4 signature. So it is quite prevalent, and its detection it could be very valuable during testing. So as I said previously, it will not improve the phenotype, but it will allow you to identify and interpret more easily this discordant CNV results when you are interrogating multiple regions. Another hybrid that is also has been described in the literature is the star 68. And this time, as you can see, the 68, which is described here in the little cartoon, has a downstream region of the CYP2D7. It actually has a small portion of a CYP2D6 that it switches from intron 1 onward into a 2D7 structure. So there is complete loss almost 2/3 of CYP2D6 sequence, which makes this completely non-functional gene. And it is often detected alongside in tandem with the STAR4. Although recently, although it's not in the public domain yet on PharmVar, but recently we have discovered that the 68 can also exist as a single unit by itself or along other alleles such as the STAR1 or STAR2. And this will be posted in the coming months on PharmVar, which will give us an opportunity to also detect these particular structures. A recent survey of samples that harbor the STAR4 this time discovered that up to 20% of these fours can actually have a 68 tandem. So although the STAR4 is defective, detecting or not detecting the 68 will not improve this phenotype, but again, it will allow you to interpret this discordant CNV results that you might observe. And increases the accuracy of your diplotype call. So one looks at the different SNPs that are interrogated by most commercial assays. If these 3 SNPs that I actually have listed here are detected, you can see that a number of alleles can be also called by the assay as long as the different structural variations are tested correctly. If this is not done correctly, all of these hybrids or all of these complex structures can be defaulted and can be all lumped into the CYP2D6 star 10 call. And this, of course, can create a miscall, and the phenotype prediction will not be accurate in these scenarios. So proper interrogation of these structural variations and these conversions that can happen either in exon 9 or upstream by having multiple CNV is very important, in addition to the SNPs that are included in most pharmacogenetic commercial assays. The other group of hybrids that are known to exist are a family of hybrids called the star 13. As you can see from this slide, there are actually multiple subtypes of the star 13. They all have something in common. In exon 1, they have this T insertion that makes them completely defective. And the upstream region here is a conversion to the CYP2D7. So in red is a 2D7 structure. But as you can see, the switch, it switches back to a CYP2D6 sequence, but the switch regions are very variable. It can start, it can switch as early as intron 1, but it can go all the way down to a post in exon 9 or downstream exon 9. So these complex structures are rare. They're not as frequent as the star 36 types of hybrids or the 68. Some of the assays that exist allow us to identify these structures if CNV are actually targeting this 5' UTR region that allow us to actually detect them. This is an example of a hybrid for which we have a reference material. For example, the star 13 in a configuration in tandem with the 2, when the switch region happens in intron 1, can be easily detected and validated. So the star 13 type of hybrids are very low compared. The prevalence is low. They were found in Africans and Mexican Americans, and they're very rare in Caucasians. And the tendency is very few actually commercial assays can detect them with high accuracy. Again, the presence of the 13 this time is very important, especially when we have discordant results, and it will allow us this time to improve the phenotype prediction by actually identifying whether we have two functional gene units or actually just one functional gene units. And I have an example of a case study where I can show you how this could be of value. There was an article in 2012 by a group at Mayo Clinic that looked actually at the frequency of misclassification due to these hybrids. And they looked at both the 36, the star 4N, the 68, as well as the 13, and evaluated the percentage of phenotype predictions before using these before detecting these hybrids. And their conclusion is that up to 20% of these samples that had the star 10 were actually hybrids of the 10 and the 36, a tandem hybrid. And up to 1% of these samples that were previously characterizing as having two copies actually had this and detected a hybrid. So this illustrates the high percentage of miscalls that can happen. And up to 7% of these had that were characterized as having duplication. So potentially having multiple functional units that can give you that ultra rapid were indeed harboring this type of star 13 hybrid, and in other words, were not actually translated to an ultra rapid metabolizer phenotype. There are multiple platforms that exist that allow the detection of these hybrids, and this slide is a comparison of these platforms. A number of old platforms, such as the Autogenomics Infinity platform, as well as the Xtagluminex platform, were not put when put on the market, were not optimized for hybrid detection. And they can all these platforms can tell you whether there are duplications or multiplications, but some of them don't give you the copy count, and in other words, will not allow you with high accuracy to predict a phenotype. But there are a number of platforms such as Veridose by Agena, as well as multiple assays that are available by Thermo Fisher, TACMan, as well as a Affymetric platform that can give you the copy count of these deletions and duplications. And some of these can also allow you to detect these hybrid structures and tandems. Some of these assays require validation, and proper characterizations of the allele counts will allow us to actually predict the phenotypes with high accuracy. None of these platforms are very effective, however, in telling you in presence of a duplication with high certitude whether which allele is duplicated. And this is something that is a limitation, and we take in account for it by doing phenotype range. Common approaches that are used to detect these variations is the interrogations of multiple regions across the CYP2D6 gene or around the CYP2D6 to allow us to detect these duplications. So for example, the TACMan assay by Thermo Fisher allows you to choose among multiple TACMan assays that target the 5' UTR region, intron 2, intron 6, exon 9, in order to detect these hybrids. But this requires understanding of the limitation of every assay, and lab directors sometimes depend on us to actually explain the utility of ordering some of these assays. And they can choose whether to run one CNV assay or multiple. In other platforms, such as the MLPA or the Agena platform, these are actually predefined for the laboratory director, and it's nice because it prevents them from actually not ordering the right one, and it allows them to actually detect multiple regions simultaneously, and therefore have a high confidence into detecting more hybrids and more complex structures. The different, some of these platforms that are used here have been actually used in the latest paper for the reference materials that were and the Coriell samples that were characterized recently for CYP2D6, I highly recommend you looking at these samples and also the performance of these different platforms, and which will allow you to order some of these reference material and be able to validate them depending on the technology you choose to run. Based on what we have looked at previously, I've selected a couple of case studies in which I can show you how the use of multiple CNV assays such as Veridose or TACMAN or MLPA will allow you to refine the phenotype and the diplotype calls. The first example is a panel, a pharmacogenetic panel, a small panel running some of the SNPs that allow us to tag, that are tag SNPs for a number of frequent alleles in a population. And this particular sample, as you can see, was positive for two amino acid changing variants, the 21,851, 4181. And by a simple look at this looks at this signature of defaulting this to a STAR2 allele, which is a normal function allele. None of these other variant tests, as you can see, were detected. So this is a sort of a type 2-like allele here. Looking at the copy number variation results, you can see that this particular patient was run with 3 assays targeting 3 regions of CYP2D6, 2 intronic regions and 1 exonic, and all of them were concordant and looked at the having 3 copies of a gene. So this will, based on this, it will automatically call for an individual having a 2 copy, a STAR2 allele with a STAR1, but having 3 copies. And because most tests cannot allow us to differentiate which allele is duplicated, we can actually imagine that the individual can have 2 copies of 1 along with 1 copy of a 2, or can have 2 copies of a STAR2 and a copy of 1. Regardless of the configuration, this individual, by having 3 copies of 2 functional alleles, is going to be an ultra-rapid metabolizer using this activity scoring method. However, if we take a closer look, if we run this particular individual for a different assay targeting the 5' UTR region or exon 1, we see now a discordant CNV results by a lower number. We have 2 copies of this individual. This allowed us to actually revisit this diplotype and by saying that this particular configuration is actually the presence of a STAR2 in tandem with the STAR13. And this, by making this diplotype refinement, we can now predict that the person is not ultra rapid by a normal metabolizer. Although the prevalence is low, as I said earlier, for this hybrid, the recommendation, the impact is very high, because the recommendation for up to 19 medications that are metabolized by CYP2D6 would have been incorrect. So there is value in having multiple regions, especially the 5' UTR and exon 1 being interrogated during this assay. The second case is, again, a small panel targeting the SNPs that are for this common and prevalent alleles. And this time, the assay did not give us a copy count, but just on a platform that tells you whether a deletion of a gene is present or a duplication is present. And as you can see, both of these configurations were labeled as part of this test as being not present. However, the sample was homozygous mute for actually the 100C2T, which is a multiple, which is found on the STAR10, but also on multiple other alleles. But by a default assignment would be a STAR10. Unfortunately, this platform does not give you the call for this particular amino acid change that actually would give you high confidence calling the STAR10. So based on this assignment, default assignment would be a STAR10, 10, so 2 copies of a 10, which predict an activity score of 0.5, which is an intermediate metabolizer. Again, if we take this sample and we run the sample on a platform that gives you copy count as well as multiple regions being interrogated, you can see that this particular sample had 2 copies for all regions except in exon 9, which indicates the presence of possibly the CYP2D6 hybrid configuration, and allowed us to revise the diplotype to a star 36 star 36. This had the consequence of refining the phenotype from an intermediate to a poor metabolizers. The prevalence of this sample is, as I said, extremely high in African Americans and Asians, and the clinical impact is visible because up to the 35 medication and the recommendation for these medications would have been incorrect. So in conclusion, discordant CNV results, so when you are interrogating for multiple regions, are extremely valuable and informative, but they're very complicated and complex to interpret. And identifying ahead the different limitations of each assay is critical before actually doing the interpretation and the reporting of these results. As we saw previously, multiple star 10 carriers that you can detect can have these complex structures such as hybrids, and therefore the diplotype, the allele that they harbor might not be a star 10, but could be a star 36 or a star 36 with a 10 structure. And this will have an impact into the phenotype prediction. Currently, although there are no standards on which variants need to be tested for CYP2D6 or which hybrids needs to be detected, we can use the frequency data and available commercial tests and their limitations to actually decide on the alleles to detect. We are part of a group within AMP and CAP that is actually building a recommendation by experts on what variants to include for CYP2D6, and we will be also issuing recommendations for the type of hybrids that are essential for detection in order to obtain standardization and have labs agree on the set of variants to detect. Thank you very much, and I will now pass the screen to the next presenter, Daryl. Thank you.
Thank you, Dr. Hachad, for that great presentation. We've heard about the importance of accurate CYP2D6 copy number determination in pharmacogenomics testing. And in particular, we've learned about hybrid alleles and how the fact that many technologies have difficulty detecting these alleles. And this leads to sample misclarification or misclassification. And it also leads to a less than ideal laboratory workflow. In the time we have remaining, I'd like to spend this time sharing Agena's answer to these challenges, and in particular, describing the capabilities of the Veridose CYP2D6 CNV panel. For those of you that aren't familiar with Agena, we're headquartered in San Diego in California, and we have locations across the world. We also have more than 500 instruments installed across the globe. Our mission at Agena is to empower precision medicine by giving laboratories a way to affordably deliver timely and accurate genomic testing. So one of the reasons our customers are able to so effectively perform pharmacogenomics testing is the technology behind the mass array system. So the mass array itself is powered by a MALDI-TOF mass spectrometer. We use MALDI-TOF mass spectrometry to analyze unlabeled nucleic acids. The basic process of mass spectrometry is that a laser provides energy to the nucleic acids, transferring a positive charge or ionizing them, moving them into the gaseous phase where they're accelerated and they move through the flight tube. And the time of flight is directly dependent on the mass of the nucleic acid and therefore its sequence. Now that all sounds quite complex, but it's really quite simple. This process happens in a very short period of time. In fact, one measurement takes around about 3 nanoseconds. So over the course of several seconds to 10 seconds, we take hundreds of measurements. which by taking hundreds of measurements in an experiment, it gives you a highly quantitative result that we're seeing on the right-hand side. With that allelic ratio of the two extended products of our primers being highly representative of the starting ratio of alleles that were present. Now, this is incredibly important when doing quantitative analyses, such as what we're talking about today with copy number variant analysis and hybrid copy number analysis. So in pharmacogenomics testing, the instrument itself provides a highly flexible format. The core consumable on the right-hand side here is our spectra chip. This spectra chip is not specific to your panel, your application. It's actually a generic spectra chip which is used for all applications. On the spectra chip is a proton donating matrix which is used to charge all the nucleic acids from all of our chemistries. We have two different types of spectra chips. There is a 96 and a 384 spectra chip. And that chip can be loaded with any combination of samples and panels. So for example, you can have some pharmacogenomics genotyping products on the chip at the same time as having CMV products on the chip, but can also load it with other panels such as hereditary genetics, liquid biopsy, or oncology panels, which provides a highly flexible and variable throughput, all the way from just small numbers of samples for those laboratories that are lower throughput, through to our highest throughput laboratories that do thousands of samples in a single day. Now, each pad allows multiplexed analysis. So each reaction can have up to 25 or more targets in that single reaction deposited onto a single pad and analysed in the mass spectrometer over the course of just several seconds. So for pharmacogenomics testing, well, of course, we have highly robust results, accurate results that doesn't require laboratories to run duplicate analysis. Secondly, we have comprehensive detection. On the same chip, you can get your analysis of your pharmacogenomic SNPs and indels, as well as a broad copy number analysis as is required for the CYP2D6 panel. And because that chip itself is just spotted with a proton donating matrix and the flight tube is a vacuum container, this does provide the most cost-effective analysis and affordable costs for laboratories that are running high throughput analyses. All right, so when we get to talking about CYP2D6 copy number testing, when we started to develop our newest Veridose CYP2D6 CNV panel, we had several criteria that needed to be met. The first one, of course, was accurate CNV calling. So we needed high concordance with current methods and high concordance with, of course, the truth. We also needed to do that detection of those complex hybrid alleles. So assessing multiple points across the CYP2D6 gene so that we can discriminate the various different complex hybrids that can be present in the population. We needed an efficient laboratory workflow. We needed a workflow that allowed you to do CNV analysis alongside your pharmacogenomics genotyping analysis, such that you weren't running multiple different workflows, multiple different platforms in order to get a comprehensive analysis of this complex CYP2D6 CNV gene. Now, we've heard a lot about the common hybrid alleles that are present in the population. And again, I'm showing you the same cartoons that Dr. Hachad showed us in the past and the complexities that can be seen in these crossovers between CYP2D7 and CYP2D6. So Agena's solution for analysing those complexities is that we look at multiple different regions across the CYP2D6 gene. So we actually look at 22 different data points across 7 different regions of the CYP2D6 gene, all the way from the 5' UTR through exon 1, introns 2, 4, 6, and 7, and exon 9 to give us a highly comprehensive analysis across the entire sequence of CYP2D6. Now, you wouldn't want to look at all of those data points manually. So what we also have is a completely automated software calling algorithm that calculates the copy number and the copy number for each of these points, and then interprets those through to the hybrid status of the CYP2D6 gene. So this is some examples of the software reporting. So what the software does is it looks at the overall copy number for those particular variants which are complete gene amplifications or deletions, but then looks at the copy numbers calculated from those 22 data points across the 7 different regions of the gene to determine the hybrid status. And then lastly, then converts that through to the functional analysis for those various different hybrids. So how does this perform in clinical samples? And I'm going to describe to you 2 different studies that we have performed to look at the accuracy and reproducibility of this new Veridose CYP2D6 CNV panel. In the first study, we ran 1,120 HapMap samples or chorial cell lines that had previously been characterized using a variety of different methods, including TaqMan and long-range PCR and other methods that have been published in the literature. And we ran these samples across the Veridose CYP2D6 CNV panel. So initially, we achieved 90% concordance with those orthogonal results, meaning that 90% of the time, the CYP2D6 CNV panel generated the same haplotype results as the prior analysis. But we also, of course, we were interested in further exploring the 10% that were discordant between the platforms. Now, the Veridose panel detects hybrid alleles that weren't analysed in various in the prior analyses that were determined that were previously performed. So we needed to employ a technique that could resolve these discordant calls to determine what the actual truth of these samples were and therefore determine the accuracy. So we reached out to our collaborators, and all of these discordant samples were then run on long-range PCR to determine an absolute source of truth. Now, when we look at the discordant samples, what we observe is that we had 13% of the overall samples having a hybrid allele, and 70% of those hybrid alleles discovered had been previously uncharacterized. So by looking at long-range PCR and matching it to the Veridose CYP2D6 CNV, we ended up having an overall 99% final accuracy by reassessing the 10% discordance. What you can see here is that the previous haplotype, which for example in this first one was a star 2, star 4, where the CYP2D6 CNV panel had, or the Veridose panel, had determined a star 2, star 4 plus a star 68 analysis, and then the independent laboratory performing long-range PCR haplotyping then confirmed that indeed this was a star 2, star 4 plus a star 68, which previously hadn't been determined by the prior techniques. So nearly all of those discordant samples were resolved and confirmed the accuracy of the Agena Bioscience CYP2D6 hybrid result. Now, those were cell lines, so we also wanted to see the performance on a large study of clinical samples. So we ran 909 clinical samples, ran that on the Veridose CYP2D6 CNV panel, and also ran it on a set of TaqMan probes. So 3 probes, one in intron 2, one in intron 6, and one in exon 9. So certainly not as extensive as the coverage in the Veridose CYP2D6 CNV panel, but giving us some coverage across the gene with those TaqMan assays. So of the 909 samples, the concordance between the 2 platforms was 98.2% compared to that TaqMan data. Almost 7% of these clinical samples possess a hybrid allele, and most of them were confirmed by the TACMan. When we look at the discordant samples or the discrepant samples, these samples were determined by Veridose CYP2D6 CNV to contain a complex hybrid that would not have been detected by TACMan because of the subselection of regions that were run by the TACMan probe. So again, good accuracy confirmed on the Veridose CYP2D6 CNV panel. So in conclusion, CYP2D6, CYP2D7 hybrid alleles are prevalent across all populations. We've heard that there are varying frequencies across the populations, and some populations have higher prevalence than others, but they're certainly present across all populations. The ability of the Veridose CYP2D6 CNV panel being a single panel, a single reaction, which gives you those 22 data points across 7 regions of the gene, allows you to analyze these complex hybrid alleles. And by using this panel, we were able to detect those complex hybrid alleles in samples that had previously been mischaracterized by orthogonal methods, and confirm that with further analyses. So with that, I thank you for your presence today, and we welcome any questions to either Dr. Hachad or myself, if you'd like to send those through.
Thank you very much, Dr. Irwin and Dr. Hachad. We do have time for just a couple questions. Our first question is for Houda. You mentioned that there are different types of CYP2D6 hybrids. What criteria should I use to decide which hybrids are important and should be detected?
That is a good question. So I think the first criteria that we use, and Darren mentioned it as well, is the frequency. By looking at the frequency and the prevalence of these alleles across major ethnic groups, that gives us a hint on the ones that we need to focus on. So that's the first thing that we do. The second criteria that it is essential to use is the availability to either clinical samples or reference material that will allow us to validate the assay, and then which increases our confidence in calling properly these hybrids. And finally, the third criteria is to really take a look at the commercially available platforms and to compare the coverage of hybrids that are currently available across platforms. And by doing just that exercise, you will see that some of these alleles that we have covered today, the examples that were shown previously, are those that we can focus on. So namely the star 36, the star 68, and the 13 in tandem with the star 2.
All right, thank you. And our final question is for you, Daryl. The presentation focused on accurately detecting CYP2D6 hybrid alleles, but it also alluded to workflow challenges. Could you discuss how the Agena solution helps with workflow challenges?
Yeah, sure. So with the Agena solutions, we have our Veridose core panels, which is our genotyping panel, and our Veridose CYP2D6 CNV panel. And both of those panels run under the same workflow and same amplification conditions, such that they can go on the same plate. So you can have a set of samples for each panel on the same microtiter plate, and that microtiter plate can go onto the same chip. So everything runs together. In fact, not just our Veridose PGX panels, but also it's the same workflow for our inherited genetics panels and our oncology panels, and they're all the same workflow, and they can all go on the same plate and the same chip, which gives tremendous flexibility. Whereas what laboratories are having to do at the moment is that they're having to run one workflow for their genotyping and then a completely separate workflow for their copy number analysis, which adds a significant complexity to the management of the laboratory. And in addition, they often have to run those workflows in duplicate, which means that they're going to have to be running many reactions under multiple different workflows to get a comprehensive result.
All right, thank you so much. We have run out of time, so I will be sending the rest of your questions along via email to our presenters, and you'll receive your answers via email. So on behalf of BioIT World's Global Web Symposia Series, I'd like to thank our presenters today, Dr. Daryl Irwin and Dr. Houda Hachad. I'd like to thank Agena Bioscience for sponsoring. And most of all, I'd like to thank those of you who came and spent this time with us. We're very grateful that you came, and we hope we were able to provide some information that will be of use to you. So on behalf of all of us here, thank you again for coming, and have a great day. Bye-bye.